Hello, my name is Asa Thibodeau. I'm a postdoctoral associate for the Uter Lab at the Jackson Laboratory for Genomic Medicine. Today I will be discussing AMULET, a novel recount-based method for effective multiplet detection for single nucleus ATAC-seq data. So what is AMULET? AMULET stands for ATAC-seq Multiplet Estimation Tool, and it is the first recount-based method that can detect both heterotypic and homotypic multiplets from single nucleus ATAC-seq data. So multiplets capture the epigenetic profiles of two or more nuclei and can either be heterotypic when they occur from distinct cell types or homotypic when they occur from the same cell type. Heterotypic multiplets can lead to errors in unsupervised clustering and differential analyses as they can form their own distinct clusters. Homotypic multiplets, on the other hand, can potentially introduce biases in allelic analysis where accurate recounts are critical. Unlike previous methods that are based on similarity to artificial multiplets, AMULET only depends on the recount information captured by single nucleus ATAC-seq data. Instead, AMULET detects multiplets in diploid cells by leveraging the expectation that at most two reads overlap a position in the genome. First, AMULET identifies all instances where greater than two reads overlap a given position in the genome. AMULET then combines these identified regions and counts the number of instances where an overlap greater than two is observed for each nucleus. These occurrences are modeled using the Poisson cumulative distribution function, and multiplets are detected by identifying nuclei that observe more overlaps greater than two than expected. In this way, recounts are effectively used to detect multiplets. To benchmark the performance of AMULET, we first estimated recall by simulating multiplets. First, we randomly paired nuclei based on cell types so that we could measure heterotypic and homotypic performance separately. Second, we paired nuclei until we generate artificial multiplets at 2.5% of the total nuclei in the sample. And finally, we combined the profiles of these nuclei and incorporated them back into the originating sample. Using recall, we compared AMULET to Archer, which is the current state-of-the-art method for detecting multiplets in single nucleus ATAC-seq data. AMULET outperformed Archer when detecting heterotypic multiplets in PBMCs, while Archer outperformed AMULET for the first of two islet samples. In the second islet sample, AMULET remained comparable to Archer. However, as expected, AMULET was able to detect homotypic multiplets at the same rate as heterotypic multiplets, while Archer, since it uses a method that cannot detect homotypic multiplets, detected these artificial multiplets seemingly at random. Although recall informs us of how well each method can detect multiplets, we also need to understand precision to measure how many false positives are called by each method. However, the difficulty to determine true singlets and measure precision prompted us to apply a different approach. For this, we turn to multiplex data from 3 donors where multiplets between donors can be identified with high accuracy when using genotype information. Using Vireo to detect multiplets between donors, we estimate precision for both AMULET and Archer. AMULET detected Vireo multiplets with 0.6 precision, while observing a low recall of 0.18. This performance exceeded that of Archer, which predicted these multiplets with only 0.28 precision, with only a slightly higher recall of 0.2. This suggests that compared to the state-of-the-art, AMULET predicts multiplets with a much higher precision. We noticed that AMULET had lower recall for this PBMC dataset than the previous ones, and determined that the lower recall likely results from the fact that the multiplex PBMC sample observed only 6,000 valid reads per nucleus compared to the 20 to 28,000 valid reads per nucleus in the previous samples. To better estimate how read depth impacts the recall performance of AMULET, we measured recall as before for samples at different sequencing depths. We observed that when the number of valid reads reaches 25,000 per nucleus, AMULET detects our simulated multiplets with high recall. In conclusion, AMULET is the first recount-based method for detecting both heterotypic and homotypic multiplets in single nucleus ATAC-seq data. Multiplets are detected with high precision, and AMULET can detect multiplets with high recall when read depth is sufficient. The underlying algorithm is fast and efficient, allowing for 100,000 nuclei to be processed with as little as 3 GB of memory and 12 hours of runtime. AMULET code and documentation is freely available on the Uter Lab GitHub, which is linked below. Thank you.